All right, third time is the charm. This is the third time I'm recording this video, and hopefully this will be the last. All right, this is our lesson on aerobic respiration. We're going to talk about what we do to break down glucose when we have oxygen around. So, this process happens in 3 steps. We've already talked about the first step, which is glycolysis, and it happens in the cytoplasm. Please note, remember, this only makes 2 ATP. Whereas when we have oxygen present, we can then go on to pyruvate oxidation and the citric acid cycle, also called the Krebs cycle. The citric acid cycle, also known as the Krebs cycle. Pyruvate oxidation and all the other steps of aerobic respiration all happen in the mitochondrion. So that's just a note. I want to also point out how much more efficient this process is. It makes 32 molecules of ATP for each glucose molecule that you're putting into this process, whereas remember, glycolysis only makes 2 ATP. So being able to perform aerobic respiration was a very big advantage for any organism that evolved this ability, and it was also a big advantage for eukaryotes as we acquired mitochondria. I will include an animation in the notes of this video, in the description of it, that will let you look at the process by which we theorized that mitochondria came to be, and let's look at this process in more detail. So, I'm going to show you this image multiple times. It is showing you on the bottom left, it's showing you the cytoplasm, and then it is showing you a close-up of part of a mitochondrion. So you see the outer membrane, the inner membrane, and then within the inner membrane you have the matrix or mitochondrial matrix, and then between the two membranes you have the intermembrane space. These are important because having these two divisions allows you to produce concentration gradients, and different reactions are going to happen in different parts of the mitochondrion and different parts of the cell. So that nice purple hexagon that we have there is a glucose molecule, 6 sides for the 6 carbons, and also because it forms a hexagonal shape. And then you'll notice we also have an NAD plus sitting out there. What is going to happen during glycolysis is we are going to take that glucose molecule, split it into 2 pyruvates, which are 3 carbon molecules. So this thing is a pyruvate. In the process, we're going to make an NADH, and we're also going to make ATP. Please note, 1, none of these things are to scale, because in reality, a 3-carbon sugar would be much smaller than the other 2 molecules. Also note, I'm not going to focus on how much of everything is produced. We're looking at what is produced where with what reactions. So let's talk about the next step. So after this, the pyruvate that's formed in glycolysis is going to move into the matrix of the mitochondrion, where the cell is going to catalyze a reaction to transform pyruvate, which has 3 carbons, as you see here, to acetate, which only has 2 carbons. This acetate gets bound to a molecule that comes in called coenzyme A, often called CoA for short. When the acetyl group gets bound to it, it forms another molecule called acetyl-CoA. Acetyl-CoA is then going to be sent on to the citric acid or Krebs cycle, where you're going to produce a lot more energy. In this process, you have 2 things formed besides the acetyl-CoA. You have a waste molecule of carbon dioxide that's going to be basically diffusing out of the cell into the blood, and then in us and other organisms like us into the lungs to breathe out. And then you're also going to form another energy-carrying electron carrier of NADH. Remember, we were saying that H stands for high energy. So let's look at this process happening. So our pyruvate is originally in the cytoplasm, that's where it's formed. It's got to get past the outer membrane and the inner membrane to get to the mitochondrial matrix. That usually happens with some transport molecules, and it's going to make its way in. Once it's there, we have a few other molecules around. We've got coenzyme A or CoA. We've got some NAD pluses, which are waiting around to pick up a high energy electron. We've also got oxygen sitting around, and what's going to happen is pyruvate oxidation is going to take that 3-carbon pyruvate, turn it into a 2-carbon group that's attached to the CoA to make acetyl-CoA, and there's going to be a waste molecule of carbon dioxide, and then you're forming an NADH. So that's happening during pyruvate oxidation. The entire purpose of this is to make that acetyl-CoA so it can be fed into the citric acid cycle. Now, I know that this is a very complicated diagram. In all honesty, it can be made much more complicated. So this is a simplified version. You do not need to know the enzymes that are used. You do not need to know the names of the intermediate molecules that are formed. The most important thing to note is that acetyl-CoA is going in and various things are coming out. You are going to pick up some high energy electrons to form NADH as well as FADH2. You're going to have 2 carbon atoms coming in to this reaction, so you need to have 2 carbon atoms leaving in the form of CO2. You're also going to use some of this energy to directly make another energy-carrying molecule called GTP. GTP is similar to ATP in that it can be used to store energy. That molecule also has a couple of other roles in the cell, which we'll talk about in Unit 4 next. What's going to happen is basically the bonds of this molecule are getting rearranged to release some high energy electrons, which are picked up by the electron carriers. And then finally, at the very end, you're going to regenerate your original substrate, whatever was originally accepting that acetyl group. So that way you can run this cycle over and over and over again. This is going to happen in the mitochondrial matrix, and this cycle takes place twice for every glucose molecule that you're breaking down, because if you'll remember, each glucose molecule makes 2 pyruvates. Each of those pyruvates is going to make an acetyl group, which is going to make an acetyl-CoA. So let's take a look at this process happening in our mitochondria. So in the mitochondrial matrix, we have a coenzyme A. That coenzyme A is going to feed its acetyl group into the citric acid cycle. Note we have the extremely simplified version of the Krebs cycle here. Remember, Krebs and citric acid cycle are interchangeable terms. And then we've got NADHs, we've got FADH2, and we've got GTP being formed. We've also got our waste products of carbon dioxide, but honestly, those are going to leave the cell, so we don't care about them too much. What we do care about is what happens to all that NADH and FADH2 we make. We've talked about the fact that they have high energy. We've talked about the fact that they're picking up high energy electrons, and we've also talked about what happens next. So we've talked about this term, we called it ATP production in our notebooks. It's also called chemiosmosis. And when we're talking about it specifically in the context of cellular respiration, we call it oxidative phosphorylation, because you're using this energy to phosphorylate an ADP and make it into an ATP. This is also the process that we performed in that activity we did on the steps, where you guys were basically taking on the roles of the electron transport chain as well as ATP synthase. So what happens is NADH is going to give its high energy electron to the electron transport chain, in the process returning to its unenergized form of NAD+. That electron, as it's passed through the electron transport chain, its energy is used to pump hydrogens across the membrane. Those hydrogens are going to build up, and that's going to store potential energy. So that potential energy keeps building up. You've also got FADH2 donating its high energy electrons to the electron transport chain, and then you're going to use that proton gradient that's built up to allow hydrogens to diffuse through ATP synthase. When they do that, you are going to use that energy of movement to form ATP. Also, a good way to remember that this is oxidative phosphorylation is just the fact that your final electron acceptor is actually oxygen. And we're going to talk about that in a moment. So this diffusion of protons back through the membrane forms our ATP, and this is all created using the electron transport chain. Let's talk about the role of oxygen now. So oxygen is very important to this process, but we haven't talked about where it's most important, and that is as an electron acceptor. So what happens is, at the very end of the electron transport chain, you need some place for those energy-carrying electrons to go. We've used up their energy, they're now low energy electrons, and we don't want them clogging up the electron transport chain. Remember when we were doing our activity on the stairs, I was originally accepting the electrons, but we also ran through what situation would happen if I stopped accepting the electrons. What happened is those electrons built up in the electron transport chain, taking up all the available space, and you were no longer able to accept new electrons, you were no longer transferring energy to protons, you were no longer making a concentration gradient, and therefore you weren't making ATP. So in order for this process to continue, we need something to accept the electron. In anything that performs aerobic respiration, that's going to be oxygen. When it accepts those electrons, it's also going to join with a couple of hydrogen ions, and it's going to form water at the end. If you will remember from freshman bio, your formula for cellular respiration is C6H12O6 plus O2 is going to yield CO2 plus H2O. We've already seen in a couple of places where that CO2 is coming out. This stage where we are getting the oxygen to accept the electrons, that's where the water is made as a product. Now, there are other electron acceptors that exist. Mostly those are used by anaerobic creatures, some archaebacteria and bacteria as well. So let's look at this process happening in the cell. First, I've added a couple of structures into our diagram. We have the electron transport chain here, and we've got ATP synthase here. Notice that we've got multiple complexes of this electron transport chain and ATP synthase scattered throughout the inner membrane. In fact, the inner membrane is chock full of these molecules. The more inner membrane you have, the more electron transport chains and ATP synthases you can have, the more energy that you can then store as ATP. So sitting around inside the mitochondrial matrix, we've got some oxygen, we've got some FADH2, and we've got some NADH. Now let's look at what happens. So NADH gives its high energy electron to the electron transport chain. The electron transport chain uses the energy in that electron to move hydrogen ions across the membrane. Once that happens and we start building up a proton gradient, those hydrogen ions will diffuse across the membrane through ATP synthase, and the cell uses that process to power the production of ATP. The same thing happens with FADH2. It is going to give its high energy electrons to the electron transport chain, which use that energy to pump hydrogens into the intermembrane space. The hydrogen ion gradient in the intermembrane space stores energy, which then transfers to the kinetic energy or energy of movement of hydrogen ions passing across the membrane, and the cell uses that energy, that kinetic energy, to make ATP. Now notice I've left those electrons in place in the electron transport chain to point out that we need to put them somewhere. We need something to accept them. And that's, like I said, where that oxygen comes in. It's going to take in that those high energy electrons, it's going to take in some protons, and in the process, it is going to form water. So now we've made a bunch of ATP. We've regenerated our electron acceptors in the form of NAD plus and FAD, and we've made some water. We're able to, our mitochondria perform this reaction over and over and over again, continuously, to produce the fuel we need to run our cells. We also use other energy sources or other molecules to power this process. We just usually have to make a few modifications before we can kind of feed it into this cycle or into this set of reactions. So what happens is any polysaccharides, any complex carbohydrates are broken down to glucose, and that glucose is then fed into glycolysis. Lipids don't go into glycolysis. Instead, lipids are broken down into glycerol and then into fatty acids, which contain most of the energy. Those fatty acids are then converted to acetyl-CoA. So they're basically broken off piece by piece, converted to acetyl-CoA, which can then go into the Krebs cycle. Then proteins are rarely used as an energy source. Basically, by the time you're breaking down proteins, you're pretty desperate for energy. But in the rare case that you do break down proteins for energy, what happens is they're broken down or hydrolyzed into their constituent amino acids, and those amino acids, depending on which one they are, can then be fed into glycolysis, or they can be fed directly into the citric acid cycle. What's the more common thing that happens is proteins are hydrolyzed to amino acids, and those amino acids are used as building blocks to make more proteins that your cell needs. There is one last thing that can happen. So you can use the energy in the electron transport chain to make ATP, or you can decouple it, otherwise called uncoupling it. And when you do that, the energy that is stored in the electrons carried by the electron transport chain can be used for another purpose. So we have a specific type of fat tissue called brown fat, or brown adipose tissue. It's not found throughout all of our bodies, and it's mostly found, it was first discovered in newborns, young babies. Also, it's found in hibernating mammals. So the purpose of brown fat is it takes the energy stored in lipids and it uses it to generate heat. The way it does this is it still uses the electron transport chain to build up a proton gradient, but instead of making protons flow through ATP synthase, it lets them flow through what's called a leak channel. That leak channel just allows protons to go through. So the energy of that concentration gradient is not being channeled into ATP synthesis. Instead, it gets converted to heat. And so that allows brown fat to generate a lot of heat. In fact, brown fat has a lot of mitochondria in it, which gives it its characteristic brown color because of the large concentration of mitochondria, and it is actually fat or fat tissue that burns extra energy. This is important for example, newborn babies, because without brown fat, they're not able to maintain a high enough body temperature because they're, pretty small. And while we do have some brown fat deposits as adults, it's not very much. It's mostly outnumbered by white fat, which is the major energy storing component in adults. And this is a potential future avenue of research. How can we try to increase production of brown fat tissue? How can we make brown fat tissue more active in adults? This might be something that could be a way to counteract, for example, the obesity epidemic. So that is the end. Enjoy your Thanksgiving break. Have a great time, and I will see you guys when we come back.